kind of a little bit of a loose lecture. I don't typically do this type of talk. I usually just talk about research, but I thought I'd put in a little background tonight about genetics. And so it might be a review for some of you, I apologize in advance, and how we use it in fisheries research. And then I thought I would go into a current study we're doing of Prince William Sound genetics in the Sound. So, by the way, I've worked in NOAA for about 25 years in the field of genetics, and that field is probably the fastest growing field in science today. I can't keep up, so I apologize if I can't answer all your questions. People want to talk about GMOs and Frankenfish and things, and I can share with you after what I know if that's your interest, but for the most part, genetics is a very fast growing field.
Do you want me to hit the lights?
Sure, at least this side, yeah. So obviously the base is DNA, and putting this together, I don't think about DNA every day in what I do, but really it's an amazing molecule when you think about it, because this molecule has a blueprint for every single living thing, and there's no two copies the same. And so it's a double helix structure, and there's 4 base pairs that it's made up of in different combinations. And adenine always binds with thymine, and cytosine with guanine. And so when you think about it, that makes it a very interesting molecule as a blueprint molecule, because if you split that in half, you can transfer that information. And this is how DNA is transferred in the cell. The double helix unwinds in a certain area of the many miles of double helix in a cell. It'll unwind just in that region enough to copy it. So this messenger RNA can take away that message to another part of the cell and produce a protein or whatever the purpose for copying that DNA is. Well, this makes it very useful in fisheries, and there's many different types of genes, right? This is actually Gene Simmons of KISS without his makeup. I thought you guys might want to see that. But really, genes in the science literature is kind of loosely used, I have found. Typically, I think of a gene as a fragment of that DNA that encodes for a certain protein or set of proteins. But a gene is really just a specific section of the DNA. And how we use that in genetic fisheries is, first we isolate the DNA, say from a fin clip of a fish. If the fish has already been sacrificed, I like to do an internal type of organ so it's less contaminated. But there's DNA in every cell, and any cell will work, right? Hair, blood samples from a crime scene. What you do is you lyse the cell with an enzyme, the outer cell wall, and you release that DNA into a free-floating solution. Then we pass it through a special filter that binds that DNA and allows all the other junk to pass through, like the cell wall, the different organelles in the cell. We're looking for just the DNA. And then we do several wash cycles where we wash, I'm standing in the wrong place, aren't I? Here we go. We wash the DNA several times, and that DNA binds that filter, and in the end, we elute it by adding a special buffer that kind of breaks down the pores of that membrane and gives us pure DNA to work with. The next step is PCR. And I don't know, I've only seen the program CSI, Crime Solver Investigators, one time, and I swear I saw that exact machine on there. So the PCR amplifies the DNA and makes many copies, because we don't want to look at the entire chain of DNA. Look how many years it took just to look at the entire chain of the Human Genome Project. Am I in your way?
No.
Okay. So we don't really want to look at the entire chain. We want to look at small pieces. Say, if it was humans, eye color or toenail length. I don't know. Some different fragment that we can compare among individuals. So how we do that is we make a small cocktail here where we have our whole DNA, We add some primers, which are known regions that flank the area of interest. Some free floating of those nucleotides that were in the first slide, A's, G's, adenine, guanine, cytosine. We put those all in a little cocktail, and then we put them in this machine, which is interesting. It heats up, 95 degrees C is quite hot. It separates the two strands of the double helix. The primers of the known region bind around our area of interest as we cool it. They start to come together, and they make 2 copies of that strand of DNA. And so we repeat this process to make multiple strands of DNA, multiple copies. And so a penny doubled every day is $1 million after a month. And so after about 30 cycles, we have about 1 million copies of the fragment that we're interested in. And now we can do something with that. It's enough material that we can run through and analyze it, which is the last step. We put it in a DNA sequencer. And there's several different things we can do with this machine, but the most common one for, say, population genetics or stock composition is we take that PCR product, the end result of those 1 million copies of our one gene, if you will, and we run it through this capillary system in this machine. The capillary system, each sample, each fish, goes through a wire there, or capillary. It's actually a little glass tube. And what we have is a known size fragment of DNA in every sample. So once it passes through, the small fragments go through first and the larger fragments go through last. And they're detected by a laser because the primers have a dye. And what we get is the output to the right. So that's 3 different fish. The top line has, I don't know, it's like a 154, 172 or something. And you see the second line down has 2 completely different peaks. And that's what would be the parents of each of those fish, right? Mom and dad. Sometimes we only see one peak, and that means both parents were the same. They had the same toe length or whatever we're looking at. And then the final fish down there you see has a copy of one of the same as the second fish, and one is a copy of the top fish. I think I have another slide, which I know it's kind of complicated to explain, but it's really pretty simple. Here's our primers on the top there in the red. We know that that sequence will target this stutter band we call a microsatellite marker. See how it goes CA, CA, CA? Well, you have microsatellites and I have microsatellites, and every species pretty much has these stutter patterns in their DNA, and we don't really know what they are used for or what they target yet anyways. In human genetics, for example, there's one area in all of our DNA that stutters like this, and we know if the stutter is really long in the 300 base pair range, you're probably going to have Hodgkin's disease when you're older. So that's the type of research going on with microsatellites in humans. But in fish, we just use it because they're different from individual to individual. And we can analyze that. So again, it goes through the capillaries, and we get this output called genotypes. That's 3 fish there. This is an example of just like one fish, one herring exactly, from Iliamna Bay. And I looked at 14 or 15 different fragments of the DNA. So you see that very top red one? That's mom and dad for that site. The second one, the black one, that's mom and dad for the same fish at a different place. So just different fragments for microsatellites, maybe eye color, hair color, I don't know. Just different fragments from that fish's DNA. Another thing that's been more heavily used recently and is definitely a fast-growing segment of human genetics, but also in fisheries, like in our lab, we use it for sockeye and Chinook work, are SNPs, and that's just a single change in the base pair from individual to individual. And actually, you need more SNPs because they're not quite as powerful. They don't give you as much information per SNP, but they're still really valuable. And we obtain them in a very similar way as microsatellites. So what we do with that data is population studies, stock compositions. So for population studies, we just tally up the allele frequencies. That is those size fragments that I was just showing you. We tally up all the 154s in that group from, say, Gravina out here. We tally up all the 172s, and we get kind of a fingerprint of what that region looks like genetically. And we do that for, I think I used 15 markers for the herring. And so you have a nice large database of genetic information. Then we look at Hardy-Weinberg equilibrium. So if you're reading a genetic paper, this is a technique loosely based on Mendel, like the pea study. I don't know if you remember that from genetic class, where he crossed green peas and white peas. And what you would expect is like an offspring of a certain amount of green peas, a certain amount of white peas, and then some hybrids. But what we do with our data, we want to make sure that it's a randomly assorted mixture, that there's not something weird, like something's under selection, or that it's a mixture itself. And that's what Hardy-Weinberg can tell us. Then we could take those two collections, let's say Gravina and herring and Iliamna Bay. And we can look at those two groups of genetic information and compare them with a G-test, which is just a kind of a probability test, if they're the same or different. F statistics is another test to tell if they're different groups. I'll show you a PC, a principal component analysis, which is more of a nice picture that lets us look at the differences between groups of fish. And of course, no genetic study would be complete without a tree that shows how related things are. But the other cool thing we've done with population genetics, I spent the first 15 years with NOAA building a salmon baseline for pink and chum and Chinook and sockeye, in which we now have baselines for the entire Pacific Rim, because salmon are cool because they're anadromous, right? So those fingerprints of those rivers are much stronger than, say, herring or other pelagic species. And once you have the salmon baseline, you can take a mixture out of the ocean and pretty much accurately predict where percentages of that collection came from. And that's been helpful with treaties with like Canada and Russia and Japan. One more technique I wanted to talk about is mitochondrial DNA, because this is an interesting thing. So the mitochondrial DNA is not part of those long double helix. The mitochondria is a small circular piece of DNA that is not even found in the nucleus of the cell. It's found out in the cytoplasm of the cell in these small organelles called mitochondria, which some of you may remember are like our energy units. And as we get older, we have less mitochondria, by the way, so that's probably why I'm tired more often, But that's where the mitochondria DNA is housed, and it's a small circular piece. And it's maternally inherited, so all of you have your mom's exact copy of mitochondria. It's not influenced by the father at all. And so it's also not as recombinant. It pretty much stays the same generationally. And we use these for long term, thousands of years, like Ice Age type studies. But we also use them to look at species ID, because typically you see small differences with non-interbreeding species. this is the type of results. This is sand lance, by the way. Do you guys know what sand lance are? Yeah. So a couple years ago, we determined all those red dots. The large circle means there's more fish with that exact mitochondrial copy. Those are all from the Gulf of Alaska and up into the Bering where they mix with the blue group, which is an Arctic species and extends over into Russia. And they are different species, and they have been, we named this new species. The other two are from Japan. So prior to this study in the literature, there were two species actually, and there's really four there by looking at the mitochondria. Okay, so jumping into Prince William Sound, I want to thank my co-authors, Heng Von Nguyen and Jeff Guyan, NOAA Fisheries, EVOS Trustees Council, Prince William Sound, Herring Research and Monitoring Program, who collected all the fish and Fish and Game, Steve. And what do I want to say about this? I guess the question is not why the herring aren't here, but why they aren't recovering. And so that was my curiosity. Why aren't the herring coming back? And my first thought was maybe they're not recruiting any herring from outside the sound. Maybe they're just trying to recover from the reduced stocks within the sound. So our questions for this study was, I wanted to look at the genetics, microsatellites, from within the sound, and look at some areas from outside the sound and see if they were different. And then two, is there structure within the sound? Do you have stocks, different stocks inside the sound? We were only able to get pretty much eastern Prince William Sound, and then this last year we were able to get some from Montague Island, which was a total score. Thank you, Fish and Game. And then some from Cook Inlet, Kodiak, Shelikof Strait, Kayak Island, and Yakutat. And then the third thing was I wanted to look at, is it structurally stable? And this might be interesting with the scale patterns, because some people think that you can tell stock structure with scale pattern, but the thing about scales is they're environmentally derived, so they might change from year to year or over time. And so that's why you need to get money to finish that. But I wanted to look and see if we saw genetic patterns, if they were the same over a few years period of time. And then three, if there were differences among year classes. Because in the Atlantic salmon, they have found differences among year classes. So we collected 500, 600 sometimes fin clips. So I throw this up here because this is what happens when you collect herring from areas of high milt. No, they don't have 4 parents. This is an example of contamination from milt. And so we couldn't really use this data because we didn't know which two went together for the real genotype. And so we borrowed a technique from University of Washington. We bleached the fins with a low dose of Clorox, a dilute amount, and it really cleaned it up nice. So, Clorox. This is the data slide I was telling you about Meadow. Not that I want you to understand like the data, but this is just an example of the Hardy-Weinberg proportions, and it's super important, and actually kind of shows our samples. We have Eastern Gulf, Kayak Island, there on the top. All the central ones there are Prince William Sound, Western Gulf, and then even some Bering Sea samples that I had. And for the most part, all of them were in Hardy-Weinberg, all the loci. And what that says to me is none of the collections specifically are a mixture of stocks. So Gravina, they all look alike. Cook Inlet, they all look alike.
Is that from 1 year or from all years combined?
That is from multiple years.
Okay.
So you see like Redhead, and I have 2 years of Gravina there. So some places I was able to get 2 years, like Stockdale. And then last messy slide, I promise. Above the blue diagonal is a G-test, and below it is an F statistic. So they're just two different ways at looking at the stocks across the board. But what I do want to point out is, notice all the groups to the right and to the bottom. That is western stocks west of the Sound, and then the Bering Sea on the bottom, and then stocks west of the Sound. All of those were highly significantly different. So Kukak, which is on southern Kodiak, is wildly different genetically than anything else. It is not mixing with any other stocks. Kamishak Bay 1 year, and Iliamna just north of there the next year, so 2 years in a row. Very similar. You notice that little white dot in the bottom? That's the only collection that's not different from each other. Kiliuta, maybe I have it wrong. Kukak is on Shelikof Strait, and Kiliuta is on southern Kodiak. And then the last four blue ones are Bering Sea collections. But all of them very different. The other thing to notice is the top row there is Southeast Alaska. So that is different. But Yakutat and Kayak are very similar to Prince William Sound. Yeah. And then two more things that stood out to me was a summer collection from Bishop Rock, which is across from Montague. Yes. Okay. And then also this past spring we got one collection of Rocky South, which seemed to be mostly 3-year-old fish, which would have coincided with that warm year from 2012. And they seem to be a little different genetically. So
it's at the entrance to the Sound, so they could easily come in from outside there.
yeah, you're right. Yeah. So we want to look more into that. There's other analyses we can do. This is just a work in progress right now. It hasn't been completed, but that is one thing I want to look at, if they are matching. But you notice they are significantly different from all the western stocks. So if they're coming in the entrance, I don't know where they're coming from.
Do you have any samples from Sitka at all?
I do.
Because in the old days, Sitka and Prince William Sound used to fluctuate together, population.
Huh.
And if it's oceanographically driven, maybe genetics would not show up, but there is always a possibility of like long distance recruitment. And did that population somehow affect Prince William Sound through immigration? So, especially if we're looking at the Rocky Bay, why that's so different. Maybe they're coming from a long way off.
Like the Alaska gyre bringing up some young.
The Canadians also have observed herring moving off into the ocean during the summer. And Doug Hay has asked the herring research about that a couple of times. Oh yeah, we saw this in BC. Is that happening here too? But we don't have any data at all from this area.
Well, that top stock there, Craig, is south of Sitka on the outer coast. It's not as wildly different as your western Gulf stocks, but it's still sticking out. I need to do some isolation by distance. Maybe it's just far away, and that's why it's genetically different. But yeah, we're starting to get some cool data. And here's the principal component. Basically, it takes all of those 15 markers and it picks just the important information and kind of shows you visually how different the groups are. And so you have all the Prince William Sound and Yakutat and Kayak all grouped together pretty tightly. That Montague sticks out a little bit, and Southeast Alaska. Oops. So there's one more piece of the puzzle I'd like to put together. Maybe I should show you the next slide first, actually. This is just one of those loci, and it's a different way to look at it. Just one of those microsatellite fragments. And each row going horizontally is a collection. So the lowest one is that southeast collection. And then it marches up to Yakutat, Kayak Island. Most of those are Prince William Sound and different years. And then up around 15 there, between 15 and 20, those are those Western Gulf collections. And you see how they're kind of in the middle between Prince William Sound and the top one's the Bering Sea. And you notice the lack of diversity, basically, in that Bering Sea collection. So let me back up. Each dot is a different size that we looked at earlier, like a 150 or a 170, how many stutters. So you see the southern fish have a lot more diversity, and all these rare alleles to the right, which stop after you leave Prince William Sound. And then you have those collections, and then there's like nothing in the Bering Sea. They're very different.
My understanding is the Bering Sea herring are very similar to the ones found in Japan. And do you have any evidence that could be a separate subspecies or different species than the herring in the Gulf of Alaska?
Yeah, I suspect they are.
So
Yeah.
then they might have mitochondrial DNA on them.
Good question. So let's back up a slide. We did. So this is a paper we put out in 2011, I think there are 10 or so. And there were 3 lineages of mitochondrial DNA. And so you're right, they do, the Bering Sea match the Japanese. This is the same pattern we saw with the sandlands, actually, or similar. But the thing to notice is that there are a small percentage of the Bering Sea lineage in the Gulf. And
contamination, yeah, going the wrong way.
well, yeah, how did they get there? That's very interesting. And remember that the mitochondrial DNA doesn't necessarily mean what's spawning with who today. This is historic, kind of, because it doesn't really recombine. It's just passed on from the mother like a football. So I wanted to marry, my next step, as soon as I get home.
Yeah, you might make it comparable to the fact that there's some Neanderthal genes in us. Everyone knows about that.
But I wanted to marry the data. I just have more mitochondrial DNA, and I wanted to marry it with this data. And I'm interested if your west of the Sound are really a mixture of the Gulf fish and the Bering Sea fish, because it kind of looks that way here to me, just from this one locus. I don't know if that looks that way to you guys, but we'll see. If it is a mixture, which I would have thought we would have seen in the Hardy-Weinberg test, but if it is a mixture, that means there's 2 species there. They're not mixing.
And by those western stocks, you're referring to the Kamishak and the Kodiak?
And the shellcock. So this is more research we're going to look at. We're going to marry the mitochondrial DNA with the same fish, and we're going to do both techniques and kind of marry that data and see what we find. So this is where we're at with the herring study. I just came back from Anchorage, and they want to look at some summer stocks, see what's going on in your sound here in the summer. Yeah. So I think that's it. Oh no. So I started off by saying genetics is a really fast growing field, and so I hope nobody asks really hard questions tonight, because I cannot keep up with the literature. So many cool things coming. One is this gene expression, and I was talking with Meadow about that before, is that sometimes we carry traits and they're either turned on or turned off depending on our environment, and not just our environment, but like our cellular environment, or with kids, their home environment. So it's that nature versus nurture thing, and there's a lot of information coming that we can maybe use some of this gene expression in fisheries. and for ourselves. I was reading a really interesting study on how people think. And if you think all negative and snotty, it actually increases your risk of disease. So, have a positive attitude, people. That's my take-home message. The second interesting area is whole genomic sequencing. Remember I said we don't want to sequence the entire double helix because that's almost too much information to deal with. But we are starting to, with the completion of the Human Genome Project, we are starting to be able to sequence larger fragments and be able to get more information. I know our lab actually has to hire a bioinformatician. I don't even know what the word is. Somebody who knows genetics and math. Because we have, these techniques coming on. And the final one, which I find so fascinating, is environmental DNA. And have you guys heard of that? Mm-hmm. Where you can take a collection of water, a water sample, and be able to determine what's in that water, in that water column by species and even biomass. So what will that do to the price of our cruises, to try and figure stuff out? They did a cool study at Monterey Bay Aquarium. If you're interested, look it up. So they know what's in the tank, sharks or whatever, and they were able to very accurately blind study, take a water sample, predict what was in that tank and the biomass of that, meaning size or weight or numbers. And not only that, it was so sensitive that they picked up what the person feeding had for lunch. There's trace like chicken and I don't know. It was a fascinating study. So very cool. I probably won't be able to get to any of this stuff in my career since I've been at it almost 30 years. But yeah, there's cool stuff coming. And it's not T-Rex. Although I thought that was, what's coming. Anyways, thank you guys. You've been such a great audience.